Good morning, everyone. Thanks for joining so early in the morning here. My name is Matt Krusen. I currently serve as the Senior Manager of Sales Strategy and Business Development at Aldevron. That's a recent title change, same roles and responsibilities. Very excited to be here today. It's been 3 or 4 years since Aldevron had the pleasure of speaking in Europe. And over those years, we have undergone significant growth, expansion, and positive change. So very happy to have this opportunity to share kind of an update on our overall capabilities with you all. For those that aren't familiar with Aldevron, we were founded in 1998 by our co-founders Michael Chambers and John Ballantyne. We were founded in Fargo, North Dakota in the US, not exactly the biotech capital of the world. However, our founders saw a lot of potential in the local culture, the workforce, land prices, and 25 years, 1,000 employees later, that decision to lay down roots in Fargo has continued to bear fruit, as we now have roughly 325,000 square feet of manufacturing space, have manufactured nearly 200,000 lots of material, and have really grown to be one of those leading manufacturers of biologics, primarily plasmid DNA, messenger RNA, and proteins. In terms of manufacturing footprint, as I mentioned, Fargo does serve as our headquarters. This is our corporate headquarters as well as home to our Breakthrough campus. Our Breakthrough campus is roughly 265,000 square feet. This is home to all of our GMP and GMP source manufacturing capabilities for plasmid DNA, messenger RNA, and recombinant protein. In Fargo, we also have our Advance campus. This is also in Fargo, but is a separate facility down the street. And this is roughly 60,000 square feet, and this is where all of our research-grade production occurs. Also plays home to our R&D team and technical operations groups. Take about a 7-hour drive southeast of Fargo. You will come to our protein facility, which is based in Madison, Wisconsin. This facility recently went through a top-to-bottom renovation and is now ISO 13485 certified. This is an R&D facility only. If there is a product that needs to be made at the GMP level, we would tech transfer that up to our facility in Fargo. Lastly, we fairly recently brought online a small outfit in Lincoln, Nebraska. This came to us via bolt-on acquisition of Nature Technology. For those that are not familiar with Nature Technology, NTC, they are the inventors of the nanoplasmid vector backbone. It's an antibiotic-free, antibiotic marker-free selection system. I'm happy to provide some more information on that if time allows. Our portfolio is divided into 3 primary business units that really follow the central dogma of molecular biology, with plasmid DNA, messenger RNA, and protein. I'll talk about those each separately here. Founded in 1998, again, plasmid DNA really was our core technology and remains our bread and butter. Over those years, we have manufactured well over 165,000 lots from research grade through clinical trial through commercial product. We currently are manufacturing plasmid in support of, I believe, 6 commercial cell and gene therapies. Of those 165,000, nearly 5,000 lots have been used in clinic or commercial product. And in addition to the manufacturing expertise, we do have in-house full capabilities for quality control, quality assurance release, and a regulatory group. The core production strategy for plasmid DNA really remains the same as you go from research grade through GMP source and GMP, easing that transition and trying to mitigate any concerns about continuity as you move into clinic from R&D. Don't want to oversimplify it, but making plasmid is biologically speaking fairly simple, growing in an E. coli expression system, lysing, purifying, QC testing. As you move into our other service levels such as GMP source and GMP, you're adding in quality systems, right? Quality review, documentation on the back end, facility and process controls. GMP source serves as our intermediate grade. GMP source really was set up to be phase appropriate for early phase clinical trial where plasmid DNA is being used as the critical raw material rather than direct API. Think viral vector or linear DNA template for messenger RNA production. In support of the viral vector space, Aldevron has developed and now offers off-the-shelf plasmids. In the AAV space specifically, we have 2 helper plasmids, as well as several AAV serotype capsids. We currently have serotype 2, 5, and 6 available off the shelf. For those that are familiar with the IP landscape around serotypes, it can be a bit challenging to navigate, but as IP allows, Aldevron will continue to bring additional serotypes online. For lentivirus, similar fashion, we have the 3 packaging plasmids available off the shelf. That we do, in addition, have a cloning backbone for the gene of interest, where we can assist the client with synthesis of that gene of interest, clone into this vector backbone, scale up in research grade, and then directly transition that into GMP or GMP source manufacturing. All of these off-the-shelf plasmids, both AAV and lentivirus, are available research-grade GMP source and GMP and are offered royalty-free through commercial. Moving to messenger RNA, we manufactured our first GMP RNA batch back in 2018, and since then have manufactured 50, approaching 60 GMP batches. At 2022 alone, really with the advancement of messenger RNA that came with COVID, we have seen that influx in our own client demand. We manufactured nearly 250 lots of research-grade mRNA just in 2022. So seeing significant expansion in that space, and we have built our facility to match. Our messenger RNA facility is capable of approximately 100 GMP batches annually. The messenger RNA that we have manufactured to date is being actively used in clinic. We're supporting phase 1, 2, and 3 clinical trials in a variety of therapeutic modalities: protein replacement, cell and gene therapy, and infectious disease vaccines. In terms of type of construct that we can support, we have developed a process that really can support a broad swath of construct types, capping modalities such as cap analogs, clean cap, ARCA, et cetera. Tailing strategies, we can either utilize plasmids with an encoded poly-A tail or can add the poly-A tail enzymatically. And lastly, we can incorporate modified nucleotides throughout the process. In terms of the process itself, we do have a process that we developed that we have seen successful from 1 kilobase to roughly 12 kilobase mRNA constructs. However, if a client does have a process of their own, which is quite common, we are happy to work with the client to tech transfer that in. We really feel that what sets Aldevron apart or gives us an advantage in the mRNA production space is the ability to source those critical raw materials internally. The vast majority of our messenger RNA clients are sourcing their linear DNA template from us. Makes sense. And this ensures the highest quality, familiarity with the material, chain of custody. It's a great de-risking strategy as well. In addition to the linear template, we offer a full suite of IVT enzymes that are manufactured in our Madison, Wisconsin site or in Fargo at the GMP level. I'll talk about those in a minute here. The last piece of the puzzle to truly offer that end-to-end solution for messenger RNA has been LNP formulation, fill finish, production of final drug product. A little bit of a teaser here, but I am happy to say that this is coming in the very near future. We are actively today manufacturing research-grade LNP drug product, and the development and launch of the GMP capability is nearing completion. We should be able to manufacture our first batch of GMP drug product by the end of Q3 of this year. Lastly, I'll touch on our protein division. Our protein services really fall into three primary buckets: gene editing reagents, messenger RNA enzymes, and a little bit of custom manufacturing as well. In the gene editing space, we have several off-the-shelf offerings for nucleases. Again, research grade through GMP. Well, technically the research grade would be ISO 13485 these days, but GMP as well. We have a wild-type Cas9. We have a high-fidelity SpyFi Cas9, and then recently worked with and licensed from Inscripta a MAD7 variant, which is a type 5 nuclease that has been branded Eureka 5. I mentioned previously, in the mRNA space, we have a full suite of IBT enzymes. Again, there's a lot of debate over what quality level is really necessary for a critical raw material, such as an IBT enzyme. The majority of the time we are seeing that ISO 13485 quality utilized. That would be our go-to quality in a GMP RNA manufacturer. However, we do have these IBT enzymes at the full GMP level and can utilize those internally or sell them externally directly. Lastly, custom manufacturing. This is admittedly not our core competency, mainly due to limitations on expression systems. But in the Madison, Wisconsin facility, which again is R&D only, we can operate in a variety of systems, expression systems, microbial, mammalian, insect, et cetera. However, if that product is going to go to GMP and tech transfer to Fargo, it has to be in a microbial system. So if that is an option, then we can custom manufacture proteins as well. In that gene editing space, we are also able to offer additional complexing services. So for duly licensed clients, we have developed a manufacturing process for RNPs, where we are able to utilize primarily our off-the-shelf nucleases, but we also can bring in external nucleases that we do not offer, such as a Cas12, et cetera. With those enzymes, we are able to do the complexing, manufacture, and release GMP RNPs in support of gene editing. With that, I will close. Thank you all for your attention. I'd be happy to answer any questions now, or we do have a small booth outside. Happy to answer questions there as well. Thank you.